I'm so sorry. Thanks very much for the hint. That was just a click too much. Ladies and gentlemen, excuse me, please. I'm starting from the beginning on. So, the broadcast is now beginning and all attendees are in listen only mode. For your information, this session will be recorded and the recording will be made available to others. The participants list as well is in private mode so that other participants cannot see your name. The chat function is in private mode as well, and only speakers and the moderator receive the chat messages. If you do not wish to continue based on the above, please log out. Again, ladies and gentlemen, hello and welcome to today's mindsON RBQM workshop. It's an initiative by Syntegrity's MyRBQM Academy. And today's talk is entitled Fraud and Misconduct Detection in Clinical Trials. I'm Tamara, the operations manager of the MyRBQM Academy at Syntegrity. This workshop will run for approximately 60 minutes today. It's an interactive session with polls, brainstorms, and time for your questions and our answers at the end. So feel free to submit questions and comments throughout the workshop using the chat box, which you find on the right side. Our speaker today is our CEO and founder, Artem Andrianov, who is a very experienced professional in the area of clinical data quality and biostatistics with more than 25 years of practical experience. So, as already said, this is an interactive workshop, and that means later I will unlock a toolbar for the brainstorming part that we have prepared for you. And in this toolbar, which you see here, I will make available for you an icon with the cup and the pens. When you click on that and next you click on that T over here, that opens a field for you, a text box that you can place anywhere on that blank sheet of paper. And here then you can start writing and actively brainstorm with us. You can even pick a different color if you want to, but you don't have to, of course. And just by the way, your entries are anonymous, so we don't see who is brainstorming what. Please don't be shy and be active in that part that comes later. Thank you very much. Good. So much for the introduction. And I would say it's time to start with our workshop now. Have fun, everybody. And Artem, the stage is yours, please.
So I hope I'm audible well and you can hear me. So I'm glad to see so many participants today. I must say that this topic always raises a lot of interest, which in my understanding is very reasonable. It's on the one hand side something which we all are aware of. On the other hand side, in fact, we want to be protected from this kind of risk and dangers as much as we can in clinical research. So I want to begin with a couple of stories in order to make this introduction to the topic. And then I would like to guide you through the question, okay, what is fraud in clinical research, how it happens, in fact, why it happens, what are the reasons of this, if there are any mechanisms and tools in order to detect it early on or be prevented in clinical trials, what particularly centralized statistical monitoring can do against fraud cases, and how this mechanism could be used in today's modern world, and why it is also exciting and important to know what this mechanism is about. So I want to begin with a couple of stories. So first of all, I myself was long years responsible for data quality in a large DC provider company. And at that days, I was analyzing clinical trials from biggest sponsor companies worldwide. And my team and I, we were always surprised to some extent to see that intentional or unintentional certain data discrepancies or data defects are a quite common thing in clinical research. Moreover, we were even more surprised that nobody tells about it, and due to different reasons, by the way. So partly because these kind of analysis is always unpleasant to hear, or the results of it. It could be connected with some reputational damage, for example, for a CRO who's conducting a clinical trial, or for a DC company who is accumulating the data. That's why usually those companies and those actors who are involved into the process, they are usually biased by default. And that's why the information about data discrepancies, data defects are not reaching sponsor companies. So that was actually a surprise for me many years ago. And in fact, that was also partly one of the reasons why my team and I were considering, okay, there is a need to create a data quality bureau, so to say, which can find certain data discrepancy and communicate to sponsors very transparently. So I want to begin with first of all with the example from the real life. So maybe many of you have heard about a fraud case which happened in countries, in African countries, in 2019, Malawi and Zambia, when there was a clinical trial on a new HIV treatment. This trial was led by a very prominent researcher, and in fact was considered to be a major step forward in fighting against HIV. The trial design was okay, and the intention was very good. The thing is, the conduct was quite surprisingly strange. In fact, healthy participants in this trial were told that they have a HIV, and in fact, they were given fake treatment. So some participants were given placebos instead of experimental treatment, and so on and so forth. So this actually, this situation or this fraud case was unveiled. The lead researcher was convicted in manipulation of the study design, and in fact, this scandal was very widespread. So we can find a lot of information about such this study scandal, which in fact put into the under question the credibility of research community in general. If you think that this kind of situation happens in African countries, you're wrong, because a year later, a pretty much similar situation happened in France with a study called GATE, where in fact it was a experimental research on a heart medication. And again, the data was falsified by the researcher. So in fact, the study design itself and the study data was falsified in such a way to look so that the results of the study look more appealing or more exciting. That was also unveiled, and that was the researcher was in fact put in jail. And nevertheless, again, that hit quite significantly on the reputation of clinical research in general. And a couple of years later, a book, Bad Pharma, appeared, which in fact shows a lot of such cases happening. So partly fight against fraud in clinical research is in fact a fight for genuine science. Yeah. So and in fact, what is clinical research? So let us first of all define the term, because in fact, while we understand fraud, in fact, in the widest in various versions, it has quite concrete examples. So fabrication, where the data is in fact is created, is made up. Falsification, where certain materials, certain data is changed or omitted, in fact, as well. Plagiarism, where certain ideas, processes, or results are kind of taken over, in fact, and without giving appropriate credit. In fact, what fraud is not including is the honest differences in opinion and honest errors. However, through long years of working in this area, I must say it's extremely hard to identify if a fraud case was, so to say, intentional or unintentional. So I would not move into this discussion just because I would suggest to focus on the so-called data defects, and they could be of a various nature. So manipulation of data, intentional or unintentional. In order to change the results, in order to so changing the study procedures that can lead to misleading findings, and fabrication of data, manipulation of study design, selective reporting of results, or falsifying patient records. So these are the usual examples. However, we will see today also that fraud cases might occur in various steps, and in fact, in various various actors might participate, and even patients themselves can be participant in manipulating of study designs and study data. So, we would like first of all to begin with the small pool or small poll of question, and we would like to ask your opinion. What percentage of clinical trials do you believe have some instances of fraud or misconduct? So please vote for your guess.
Thanks very much. I can see the answers coming in. Great. And I'm presenting the results now. Thanks very much for participating. So here we go.
So I can see here that the opinion percentage has almost every answer has got pretty much similar part of so with dominance on 5 to 10 and 10 to 20. So in fact, and the answer with so the second answer with letter B, in fact, has the major amount of answers, and based on publications and our internal research, we tend to believe that approximately 5 to 10% of clinical trials are affected through certain types of fraud cases. Based on publications, you can find various estimations with 2% of proven fraud. The problem is that in many cases, in majority of cases, it's extremely hard to prove, and that it is a fraud situation. And in many cases, as I mentioned before, certain data discrepancies appear unintentionally. So nevertheless, based on long research internally by Integrity and communicating with various opinion leaders in this fields, I can say that in many internal studies in pharma companies, they come to a conclusion that around 10% of trials were found to have significant reporting errors. Yeah, so let's move forward. At the same time, I must admit that for a pharma company, even one case through the whole history could be absolutely damaging. So the damage of reputation that happens through this situation is devastating. In this case, even if we speak about 10%, which is a huge number by default, but within each pharma company, you might have heard, so to say, situations where researchers were aware about certain misconduct. So based on Duke University analysis, more than 40% were aware or are aware about certain misconduct but did not report it. A large number of scientific papers were retracted in the recent years. And in fact, that's also one of the signals that we have to work deliberately on improving the genuinity of research and verifying, so to say, the results before the publication several times. So this is, in fact, that's what we're supposed to do. So again, intentionally or unintentionally, we suppose, so we need to know that errors, misconduct, data defects, and ultimately fraud cases happen in clinical research. The question is when, and in fact, how are you prepared for such situations? How are you prepared to detect these problems?
And Artem, please excuse before we continue. Perhaps we can answer a question from the audience. Can you speak to the types of studies? I presume treatment indication may be important consideration.
Yeah. Very good question. In fact, types of studies in this case, so I would say for example, in the different phases of studies, in different types of studies, different misconduct happens. So if we speak about early phases of studies, for example, we are more seeing, so to say, the patient-driven misconduct, where patients patients receive monetary gains out of this, and professional patients, so-called appear, who kind of try to misuse the system. So different indications. Of course, I would not expect saying fraud cases in oncological studies. There's more unintentional errors. However, as I mentioned before, HIV studies or cardiovascular studies, in fact, are quite affected, where there is a lot of patients, there's a lot of information, and in fact, to conduct such data manipulation is way easier in comparison, for example, to rare diseases studies or cancer studies, where the amount of patients, the number of patients is way smaller. And in fact, particularly in rare diseases, each patient, in fact, is very, very well known, so to say, for a research group. So I have not seen any spread between fraud cases and types of the study. Of course, the biggest the study is, and the one I mentioned before, the Gate study, for example, was quite significant in France. Of course, these scandals are well known worldwide. Nevertheless, in smaller investigator-driven studies, for example, we as a company have seen a lot of misconducts and a lot of errors. Sometimes they happen due to lack of time and resources to check and verify the results. And this is where, again, some automatized mechanisms could be of a big help. Okay, I hope I have answered the question. If you have follow-up questions, please feel free to write additional, and again, we can address it on the fly. So, why fraud cases happen? Very complicated question, because if we understand the motives, we can also work dedicated. So we can dedicate our efforts to reduce these motives and make it not so attractive. And what are the examples and what are the ways to prevent such instances? So why fraud and misconduct happens? So there are many internal motives that drive this situation. Of course, you need to receive a desired result, one of the significant. So as a researcher, usually you work 10, 20 years on certain medication. And of course, whole your being is dedicated to this project. And of course, when the results are not as expected, there is a big desire to adjust the result, to reach statistical significance, for example. There is a certain effect, which I've heard many times speaking with pharma companies. For example, if a clinical trial fails, nobody wants to repeat it. So you have more or less, as a researcher, one shot in order to qualify or to check if the medication works or not. And just imagine CRO, for example, is not quite well chosen, and CRO is not doing their job good enough. So statistical significance is not reached. So there is a very small chance that there is another opportunity. So for a researcher to receive the results. And in fact, that's a big motive as well. So to reach statistical significance, despite on the certain data variations that happen due to accumulation of information. Of course, monetary gains. So we were speaking about phase 1, where patients receive monetary gain out of clinical research. And of course, some of patients need to stay at home in the hospital during this period, but some of them leave the hospital, and there is a big desire to get to, in fact, to maximize the profits, so to say, by certain individuals. In many cases, we saw professional patients, in fact, as participants in the later phases of studies, phase 3 as well. Certain sites were telling us that there are companies, in fact, where you could book and order so-called patients. You can call and order 20 patients, for example, next day. So they will appear in your doors. And in fact, if a site stays under pressure in order to deliver certain results, sometimes there is a big desire to use such kind of services. Of course, we heard a lot about such services, for example, available in Asian countries, for example, where literally there's opportunity to get a certain number of patients, whatever is required. Sometimes just ignorance, naivety. So in fact, that's also how to deal with data, how to accumulate it, how to evaluate it. That's also a very, very big part of research, and certain carelessness and sloppiness. In fact, that's where certain fraud cases appear. So if you think again, that's something which we have left, so to say, far behind us, and now in current modern days with EDC systems, with professional CROs and certain auditors, internal auditors, external auditors. If you think that is far in the past, in fact, you can see just a recent publication where a Florida woman was jailed for lying to FDA about children's drugs trial. So this publication from in January 2023, in fact, so about big fraud case, which happened a couple of years ago, nevertheless continues with a sentence here. So that was a children trial with asthma, and in fact, doctor has manipulated the data there. So the consequences of fraud case are always very devastating. In fact, if it was detected or not detected, this literally doesn't matter. Or it matters, in fact, for the company, of course, but in fact, for the genuine research, scientific research, both situations are very disappointing. So if the fraud case gets unveiled by FDA, by EMA, or by whistleblower within the organization, then corporate reputation could be nearly extinguished or literally damaged. And it's extremely hard to measure the amount of reputational loss that might happen. So this in large organizations, very quickly it can turn to billions of loss just because of the reputational loss. There are direct financial costs of connected with recalls, with retracting of publications, and providing additional information to the public in this direction. But of course, again, as I mentioned in the very beginning, the whole clinical research suffers from such cases. And that's where we feel obliged to fight against that, in fact, with all our passion. So there is a negative impact. So, for example, after these trials, which I mentioned in the African countries, there was a big loss of number of patients who are willing to participate in clinical research. So, in fact, the problems with recruitment, which are hard to connect, so to say, with fraud cases, they are connected. And in fact, patients are thinking, okay, we don't know what can happen with us. So we remember there was a study in Würzburg when some healthy volunteers were given an experimental drug, and a lot of them suffered. So this experimental drug caused severe side effects. So that's why nobody remembers when it happened. Nobody remembers, okay, what was the reason? But these scandals left in the public memory for many, many years. And in fact, that's where reasons in the certain troubles of recruitment. So that's exactly what the side effects, so to say, of these situations. And I see some question which we may address.
Yes, there is in fact one question. Wouldn't the unreasonably complex study design add some risk for the wish to fraud when such design ends up to be unable to answer the scientific questions or study endpoints?
Yeah, I love this question. It's very, very aligned with many of situations which we see, in fact, in clinical trials right now. So sometimes unreasonably complex study design is part of the problem as well. Yeah. So it, of course, increases the number of backdoors a number of troubles, intentional or unintentional, that can happen through the conduct of study. So that's a part, so to say, of the problem, in fact, or part of the side effect, where clinical researchers design a study in such a way that is extremely complicated to navigate through the data, in fact, extremely complicated to navigate through the protocol design itself, and later to evaluate if this result is trustworthy. So, and that's where, of course, this is again as part of the, I would say, as a wish to fraud, or part of the hidden strategy, so to say, through these studies, that we kind of observe. Good medications usually, in fact, do not require complex designs. They do not require additional data, which will be not looked through and evaluated. So these complex designs are partly due to history or scarcity against the regulatory bodies, and in fact, many pharma companies, as they have to invest certain years in a clinical trial, start to accumulate way more information that is required to prove if medication works or doesn't work. So I truly believe that simplifying protocols, truly believe that simplifying study designs is a big step towards reducing the possibilities of intentional and unintentional fraud cases. Okay, so here I need to really to guide you through these steps. And in fact, as clinical data get accumulated, many actors are involved. These could be sites, patients, CROs, and those teams who are responsible for data evaluation, data exports. And on each step, certain misconduct and fraud might occur. So manipulation with the protocol, misuse of measurement tools, calibrational problems, these are usually troubles which we see very commonly on sites. During the accumulating the data, so to say, patients and professional patients are those who could affect the study, and in fact, very commonly, professional patients are working together with site personnel. So sites are aware about these patients. They are calling them deliberately when they need certain recruitment rates or improvement of recruitment rates. And again, these patients come in, either they are not taking medications, or, which is worse, even taking several medications and participate in several studies simultaneously. So this is where, again, how big is the problem is very hard to estimate. However, based on our estimations, a lot of studies are affected through that, and particularly, so to say, where moving to certain countries, it automatically kind of opens this Pandora box, and this is where we need to be very aware that this situation is something to be protected from. In fact, big actor in the study conduct is our clinical research organization, whom we trust, in fact, the conduct of the study, in fact. And these organizations, again, intentionally or unintentionally, very commonly are affected through certain errors, and they are not willing to communicate these errors openly to sponsor companies. Sometimes it's connected with, again, being scary of reputational loss. Sometimes it's just being unaware. And we had situations where sponsor companies decided to switch CROs in the middle of the study, or were notified that in fact studies really is not running at all after a year, when CROs in fact was not doing any work. So such situations always surprises me, in fact, and I must say your CRO is a partner in conducting the study. In fact, and the vendor oversight, to my mind, should be based on transparency, and this transparency ideally should be built in independent way. But even when the data is accumulated, the export itself can contain certain errors. So the evaluation of the results, of course, here the controlling mechanisms are more rigid. Nevertheless, in my experience, I saw cases where a certain amount of data was unintentionally exported, and a big chunk of information was not exported by mistake in the exports. And of course, depending on the difference in the results, that could be a big or not so big problem for pharma companies at the end of the day. So don't forget that usually, even if data export is validated procedure, nevertheless, validation happens in the different environments and different circumstances, and with different amount of information. That's why many data defects which happen in this stage are happen due to just amount of information that's connected with this. So what this slide is actually supposed to demonstrate is that you need to be aware about the pipeline of data accumulation, and verify each step and put the controlling mechanisms on each step in order to get notified about certain data defect cases.
So, and here we are coming to the brainstorming session that I mentioned earlier. Artem, what question do we have for our audience?
So we would like to ask you for what is your opinion, what steps is your organization take to detect instances of fraud and misconduct? So if you could share with us your mechanisms, your steps, your examples, that would be really great for the whole audience.
Yes, and to do that, just click on the icon with the cup and the pens. Next, click on the T, and you can start writing. What steps does your organization take to detect instances of fraud and misconduct?
So don't be afraid to share this information. I see
Click on the T box, please. The box with the T, yes, great.
Mm-hmm.
Ongoing clinical sense data review is coming here as a comment in the chat. Monitoring. Thank you very much. That looks good. Yes, thanks a lot for your comments. So, Artem, let's see what we have, please.
SOPs. So great. I see a lot of very exciting results and exciting steps. And I must say that there are some mechanisms which are rather happen before study starts. For example, conducting background checks on all research personnel. That's something which could be undertaken as part of SOP, and I see some SOP here as an example. There are some mechanisms which are happening during the study conduct, and I see a lot of examples here, like central monitoring, statistical monitoring, site monitoring, regular control and verification. So routine developed to identify suspicious data too good to be true, trend review, KRI review. So this is very important to understand that certain steps are mitigation steps towards to understand, or to, in fact to prevent or to reduce, not prevent, but reduce the this risk. So in background checks could be already as a good preliminary step. When the data gets accumulated, very important is to start reviewing the inconsistencies, anomalies, and encourage, in fact, a culture of transparency. And I think that's a very important, which kind of mechanism and tool on itself. Yeah. So there is a famous saying of economist Potter, who said, "A culture is a strategy on breakfast." And what he meant, in fact, is that when we establish a culture within an organization, then every strategy will either work or doesn't work or will not work. So, in fact, culture is a big part of dealing with fraud case. So transparency, open communication among researchers already assist and help you on this part. But also, of course, looking into the data, identifying the anomalies in the data, it's part of these elements, part of this strategy. So, and this is in fact where what we want to promote and address today. Somebody has found pens on the fly. So to detect the various anomalies and inconsistencies, so organizations supposed to set up a chunk of risk indicators, key risk indicators. And these key risk indicators, these are like a small smoke detectors, which in fact sits nearby to each process, like activation of site, first patients, inclusion criteria, protocol execution, monitoring, and they warn project management about certain issues in inclusion-exclusion criteria, SAE reporting, or adverse events reporting. So that's where you start already getting warning signals on the fly, so to say, if something systematically goes wrong. So in order to establish these risk indicators, again, you're supposed to understand what risk you're going to control, and to document these risks in advance. So and that's a very important and essential part of it. Understanding the risks, understanding what risk prioritization are you going to undertake here, and what risk indicators will help you to detect these risks. If you will look on the ICH guidance and we go through the risk management procedures, which consist with from risk assessment, mitigation, risk issue management, we can infer that, in fact, centralized statistical monitoring is one of the elements, one of the essential elements in the detecting the anomalies and detecting those data which is different. And why it is so? Because again, centralized statistical monitoring can, for example, compare all data points together, patients with patients, site with sites, countries with countries, and in fact identify those patients, those sites, those measurements which are statistically different. And difference is exactly the hint towards detection of anomaly and fraud cases. So, in fact, deviation from the norm is your main driver to understand what you're looking at. So I would like to give you a couple of examples how it looks in the real life. And I'm using the examples from real studies where me, myself, and our team was involved in order to identify such cases. So what you see here is an example of comparing all the data points for various patients. So these are the patients were mapped in a big heat diagram. So and every data point in the measurement was compared to each other. The scalar here was used in order to identify those elements which are similar to each other. Again, when you're looking for deviation from the norms, sometimes you're looking for those data which is too good to be true. And this is exactly where the similarity comparison helps with. So similarity in this case was highlighted with red color. So if, and it was measured in percentage. So it's not exact match, but it's similar values. And when we looked deeper into this study data, we identified those subjects and where these measurements and with different subject numbers were exactly the same. So sometimes they were very near to each other, but in many cases were exactly the same. Unfortunately, this site has accumulated way more data than the others. And unfortunately, particularly this site, has influenced the study results in such a way that the whole study failed. So again, that was the case where we didn't call police, but with very high probability, we have considered that due to certain misconduct and these elements and accumulation of information, the whole study suffered and the whole study failed. So when we look into the various measurements, it's of course also possible to detect certain hints towards misconduct. And for example, comparing the ECG signals between patients. Yes, it is a complicated way of dealing with that, but with automation, in fact, it using of currently with artificial intelligence and machine learning mechanisms, it is possible to detect the similarities even within ECG signals, similarities within spirometer signals. And this similarity could be investigated further on in more detail by a professional who can identify if it's really the copying of information or not.
Artem, please excuse. I would like to clear a question from a participant here to the values before. What did these values tell us and why were they similar? Perhaps we can answer that before we continue.
Mm-hmm. So I assume it was the question in the regarding this slide. So these values were measured in different visits for different subjects, and they are absolutely identical. And were measured within one site. What it tells that with very high probability, this site conducted a misconduct in this case. So copied the information from one patient to another. And that's where, with a very high probability, if the information was copied, so to say, from those patients where for example, the medicational effect was negative, then this side affected the whole study in a negative terms. So that's generally, so this is an example of fraud case by creating, so to say, of data and showing this information, in fact, through the submitting this information to sponsor a company. Of course, manually to detect such cases, it's almost impossible, because here we are dealing with a whole huge number of computations where there is a need to compare everything with everything. So, and if you remember from mathematics, it requires a factorial of calculations. So these are hours and hours on the server side, so to say, in order to detect such cases. However, when they are detected, then you could, as a researcher, identify very precisely if something deserves attention, or is it just a case of coincidence. Okay, understanding the, of course, the curves or the measurements, the way how they're supposed to be done. For example, in spirometer case, there is a ATS criteria check. So from American Thoracic Society, where there is a certain criteria needs to be found from one efforts to another, like repeatability. So understanding these kind of details is very helpful in detecting the anomalies. So, and very helpful in detecting the wrong measurement types. I remember a case in spirometry when, for example, the measurements were taken in the African countries, and they were some of the measurements were taken in the morning day hours, and some of the measurements were taken in the afternoon hours. But the difference in temperature were so large that equipment supposed to be recalibrated. That was not done, and that was also affected the results at the end of the day. So understanding also in what circumstances the measurements is done is also very important. So we have 8 hours or 6 hours to, in order to kind of come to final steps.
Yeah. Not hours, but minutes.
For minutes, yes.
Hours would be great, wouldn't it?
Yeah. So how can we deal with that? In fact, I think the rule number one is adopting the zero tolerance to all misconduct cases, and implement it, as I mentioned, in the form of culture of transparency and open communication within the organization, and building the mechanisms which informs project management team, sponsor, without conflict of interests about the cases, about the problems, so to say, in the data quality. So applying centralized statistical monitoring methods across study, or even across the whole program of studies, is very beneficial. And using this power, so to say, of comparing what is norm, what is deviation from the norm. So that's what is definitely a very powerful mechanism. Continuous data sanity checks, rules engine checks. So these are the mechanisms which are, again, applicable. Nowadays, we see more and more examples, and very good examples, of application of artificial intelligence, machine learning, in order to detect trends and anomalies across different studies. So again, this is something which is very reasonable to explore. We have here certain algorithms, and I'm going for the sake of time, I'm going to skip these slides, but please be aware that these mechanisms, these algorithms are described in the slides which you will receive after this webinar. I want to say that all these algorithms which are listed here were proven by Syntegrity and other researchers to be very useful in fraud detection. And that's what you can take based on publications, for example, and apply in your clinical research after this webinar. So we do advise to take a look on various digit preferences algorithms, for example, which measure what digits are preferred by a certain researcher. Trailing digits, leading digits, which can show that the site is measuring genuinely or measuring artificially. And this is where already good results appear. So one of the famous digit preference algorithms is called Benford's law, which in fact expects that there is certain distribution of numbers in data which is accumulated by summing up certain values. So you should expect, for example, more ones or twos rather than eights and sevenths in distribution of information. And that's where Benford law is applicable. It's not applicable for all measurements, but applicable for those measurements where for many real-life sets of numerical data. So these are, of course, through this law is applicable for the information which occurs through collecting certain informations. And this is an example of one situation, let's say, where this Benford's law for certain site is extreme. So it means that, as you can see on an orange line, so a blue graph shows how the data digits supposed to be distributed. So ones, twos, threes, but you can see a big jump under 7. Again, this is a signal to see what is going on at this particular site. So, and then this other sites were quite much better in the distribution of digits. So, uniformity check, very good algorithms also to where everything is compared with everything, so to say, and by checking the trailing digits, it definitely helps to identify the deviations from the norm. So, in interquantile ranges shows, for example, the outliers. But even simple things like scatter plots help you to identify the anomalies very quickly, and data distributions can assist you with that as well. Correlation matrix shows you the correlations between different data chains. And in fact, this is where, of course, it's also important to notice certain correlational discrepancies. But even simple histograms can give you already a hint where to look if there are some certain outliers happen in data sets. Again, I'm skipping this information deliberately. You can take a look on the slides which we provide.
Yes, and here we have a question, please, again for you. What do you believe is the most common form of fraud or misconduct in clinical trials? Is it fabricating or altering data, falsifying study results, misrepresenting the safety or efficacy of a treatment, or perhaps recruiting ineligible participants? What do you think? Thank you all very much. Looks good. I'm presenting the results. Great. Thank you.
Yeah. So I see that fabricating or altering the data together with recruiting ineligible participants are getting the most of attention from the audience. I personally also agree with that. This is definitely an area where a lot of clinical trials suffering. So fabrication happens on different steps, so to say, and altering the data by researchers, by sites within CROs, and later even and in recruiting ineligible patients, in fact, as we discussed already before, is also something which clinical trials suffer due to the need to recruit patients. In fact, in certain times, quick at sites are under pressure in order to conduct them. So I would like maybe to say a couple of closing words, and I would like to thank everybody who participated in this webinar today. I know that this topic is very complicated and very sensitive. However, I would like to appeal, as I mentioned before, to accept zero tolerance to that, accept or promote culture of transparency, conduct unbiased evaluation of information regularly, react on that, do the mitigation actions, corrective actions, and preventative actions accordingly. And I think you will be in a very good situation, very good, safe, on the safe side in regards to the fraud and misconduct. Thank you very much and appreciate your attendance.
Good. Thank you also very much, Artem. I would like to address one more question from the audience. How much time do you spend on reviewing noise flag caused by comparing everything to everything for middle-sized studies?
Interestingly, when the data is compared everything to everything, so to say, then the noise amount gets quite significantly reduced. So what we are looking at is not individual escalations, rather we are looking at the systematic escalations. So if there is some data points which are similar or the same, it's not what attracts our attention. Where we are rather concerned if we see, particularly by one side, by one investigator, by one professional, particular systematic errors where certain data looks very, very similar to each other. That's what it attracts our attention. And this is usually, there's not much noise around it. So this is the cases which quite easily, after the research is conducted or after the calculation is conducted, is easily visible. So in this case, but of course, the size of the study is definitely important. Yeah, so if you have 12 participants and you compare everybody with everybody, of course, the amount of noise will be higher, rather than if you have thousands of patients, for example. So that's definitely also an important criteria. Yeah. And that's why we were referring to that sometimes comparing to other studies within a program or group of studies, in fact, is also relevant and important as well to reduce such noise cases.
Thank you very much, Artem. I could imagine there is a lot more to say about this topic, fraud and misconduct detection in clinical trials. So, ladies and gentlemen, what do you think? Should we perhaps have a second part of this mindsON session here on this topic? Oh yes, I see the responses coming in. I was just going to say, please then write a yes in the chat. Okay, great. And the more yeses that are coming, the more sure I am I will set up a second, a continuation of this session, so to say. All right.